Thank you for the opportunity to share with you my research interests. As Gleb mentioned, I'm in Naples, Italy, and I'm a researcher on multimodal imaging for the study of brain connectivity. Let me just shortly introduce myself. I'm a physicist with a PhD in computer science at the University of Naples, Federico II. And I performed my postdoc at the Functional Instrumentation and Imaging Group in Pisa, Italy. And since 2012, I'm a researcher at IRCCS SDN in Naples. Just a short introduction about my institution. IRCCS stands for research institute recognized by the Italian Ministry of Health. In particular, we are recognized for integrated diagnostics. And our mission is to promote research activities in integrated diagnostics, in vivo and in vitro diagnostics, to transfer research to clinical practice and market, and dissemination and training for students and professionals. We belong to several national and international networks, such as we belong to the multimodal molecular imaging node of Eurobioimaging, IATRIS, national networks of clinical institutions, EIT Health, and the BBMRI network for biobanking. And I'm in charge of the coordination of the laboratory of image processing, and I'm collaborating with the laboratory of bioinformatics, 3D printing laboratory, and also biobank. This talk is organized as you can see in this slide. The first block of slides is about multimodal imaging hardware that is strictly related to brain connectivity data acquisition, a description of brain connectivity approaches and data processing, as well as some clinical examples and remarks about how to exploit multimodality in brain connectivity studies. Just about hardware, we have the main modality for clinical imaging are computed tomography, positron emission tomography, and magnetic resonance. On the top, you can see a picture describing PET imaging. Essentially, PET scanner is a detector of a radio tracer. Therefore, what we can see is the distribution of the radio tracer. PET scanner strictly depends from the administered radio tracer. And PET scanners was powered with a CT scanner, with the first hybrid multimodal imaging of PET-CT, and PET-CT allowed faster PET scans with anatomical reference. But it was, on the bottom, you can see a scheme of a PET-CT scanner. It was a sequential scan. All the PET-CT scanners are sequential, and you have to acquire first CT and then PET imaging. In 10 years ago, technological development released the first hybrid imaging modality simultaneous. PET-MR was indeed simultaneous, as I will explain here. Technologists had to face some challenges in order to integrate PET within a MR scanner. Both are very complex technologies. Here you can see a list of challenges. And 10 years ago, Siemens released the first PET-MR scanner. Here you can see a scheme. We in Naples we installed our scanner as a pioneers in Italy, and the scanner is like a magnetic resonance. But if you go within the scanner, you can see here a slice of the scanner where PET detector rings are embedded within a scanner. The magnetic resonance scanner is equal to a Vario 3 Tesla scanner from Siemens. This is a short chronology of PET-MR scanners release. The first was installed in Munich. The first Italian PET-MR scanner was installed in Naples 10 years ago, as I said before. We experienced some major update of the scanner with some important improvements. 6 years ago, the first PET-MR scanner based on silicon photomultiplier technology was released by GE with other major updates for the biograph. And I have a statistics of three years ago. We have about 100 integrated PET-MR scanners worldwide. This is a map of the scanners. In Italy, we have 4 installations from the north to the south of Italy. There are still open issues related to PET-MR scanner. Here you can see a list of the open issues. The major challenge related to PET-MR scanner is the attenuation correction. Therefore, for PET imaging, you need to consider the medium that is where the photon from PET radio tracer travels, and attenuation correction should consider the attenuation of the human body. And with MR, you can have just a surrogate of the attenuation, and you can have an idea of the importance of this topic with this graph that is a query from PubMed. And you can see that from the first technological development of PET-MR, we have a high increase of attenuation correction related to PET-MR topic toward 400,000 citation during the years. Another related to the attenuation correction is that everything is developed to work with PET and PET-MR should be transparent with respect to PET imaging. Therefore, for instance, a EEG cap should be validated to be transparent with respect to photons traveling from PET radio tracer. This is crucial to have a PET EEG MRI, since another multimodal imaging modality very important for multimodal, for brain connectivity studies is indeed EEG MRI. Some developers released, I think that there are 3 or 4 products worldwide of EEG that is completely MR safe and could be performed during fMRI and structural MRI acquisitions. Here you can see a scheme of the complexity of EEG MRI. You have to bring out of the gantry, MR gantry, the amplifier, and all the information should be moved through optical fibers on the console with all the acquisition paradigms. And on the bottom, you can see me and my colleagues during our test with EEG fMRI. In our case, we have a 96 channels high density EEG fMRI. Toward fully integrated PET, MR, and EEG scanning, we have also my colleagues from PISA worked as a coordinator of the TRIMAGE project. In PISA is in the center north of Italy, in Tuscany, and they, I think that 1 or 2 years ago, they released the first trimodal scanner, the so-called TRIMAGE scanner, that is cryogen-free, is a 1.5 Tesla magnet, and is fully dedicated to the brain imaging. Therefore, it is fully designed for brain connectivity scans, and will be installed this year in PISA, the first prototype. This is a famous picture about integration of MRI, PET, and EEG scan from an article from the group of Ulick, Shah et al. And this picture shows how each modality complements each other. Therefore, we have several features, the ability to scan anatomy, molecular imaging, sensitivity, specificity, clinical utility, and function, temporal resolution, spatial resolution. And here you can see how is strong the complementarity of MR, PET, EEG, that essentially close the picture with high effects on all the features with the full integration. Therefore, with such kind of instrumentation, such kind of multimodal instrumentation, we are enough, we have enough power for acquiring brain connectivity data. Therefore, let me explain what we mean with brain connectivity and what are the approaches in literature for brain connectivity. Therefore, as I said before, multimodal imaging can give a multimodal window on the brain. Here you can see some low level and high level physiological effect of neuronal activity. You can see on the right neuronal activity, and related to neuronal activity, we have uptake of glucose, increasing blood flow, oxygen demand, direct the displacement of deoxyhemoglobin, ATP consumption, going upper, integration and signaling in ensemble of neurons, and sensory, motor, and cognitive processes. On all such kind of aspects, we can probe with different modalities. Here you can see, for instance, with resting state fMRI and DTI, we can study the integration and signaling in ensemble of neurons. With for instance, magnetic resonance spectroscopy, ATP consumption, and astrocyte. With FDG PET, the uptake of glucose. With arterial speed labeling or oxygen PET imaging, the uptake of increasing blood flow or uptake of oxygen, and displacement of deoxyhemoglobin with a Bold signal. And with EEG and MEG, we can directly probe the neuronal activity. Therefore, you can have from this an idea of the power of G, PET, and MR imaging. And the simultaneous imaging can give useful data for studying also the relationships between different aspects. Therefore, we can use PET-MR EEG imaging to study the relationship of FDG PET with the functional connectivity, for instance, or EEG with the resting state fMRI, and so on. Regarding brain connectivity, that is our main topic today, here I'm reporting the definition of brain connectivity during the first article of the journal Brain Connectivity, that is a journal released 10 years ago. And in the first article, Brain Connectivity, a new journal emerges, we have, I think, a nice definition that brain connectivity research is concerned with anatomical pathways, interactions, and communication between distinct units of the central nervous system. And very important, we can have different approaches that study brain connectivity at different levels, micro, meso, and macro scales. And very important, we are constrained by the limits of temporal and spatial resolution in modern in vivo imaging techniques, reporting all the techniques that are object of this talk. Therefore, it is very important. We are studying some surrogates of actual brain connectivity, but with very insightful for neuroscience. Here you can see another PubMed search that was related to 4 different keywords: brain connectivity, connectome in red, in yellow functional connectivity, and in green structural connectivity. All these keywords will be explained during this talk. And as you can see, there were an increasing a rising publications, citation curves during the years, starting from year 2000. And this is very important. As you can see, functional connectivity is the most cited, but also connectome and other aspects of the connectivity are highly cited. As I said before, we have different approaches for brain connectivity, and we have first all the most intuitive connectivity approach, and is anatomical or structural connectivity. That is indeed the effective presence of axonal connections between brain regions. Therefore, this is an effective linking with axon between neurons and or brain areas. But if you go through the literature, you can see also related to anatomical or structural connectivity concept, also the study of the structural covariance. I will explain the covariance across subjects for metabolic connectivity, but it could be applied on also structural and functional connectivity. The second approach is functional or so-called intrinsic connectivity. That is indeed the statistical dependencies between a regional time series. And with functional intrinsic connectivity can be probed with a resting state fMRI, resting state EEG, or with magnetoencephalography. And we have also several approaches for data analysis. We have a seed voxel, aggregate decomposition, or independent component analysis. The third is effective connectivity. That is indeed the study of causal influences between neurons, also called the direct connectivity. And in this case, I want to know where the neuronal signal starts and where it arrives. And this is strictly related to the temporal resolution of our imaging scan or imaging apparatus, and considering the high temporal resolution of EG and MEG, such techniques are strictly related to EG and MEG, but we have to consider also that how accelerated sequences are under development for fMRI, the so-called multiband imaging, and also fMRI can be used in order to infer effective connectivity. The fourth concept is metabolic connectivity, that studies statistical dependencies across subjects related to PET-acquired imaging. The majority is related to fluorodeoxyglucose PET, so-called FDG PET, but you can check on online that other approaches that employs several and different radio tracers can be found. And here I'm reporting an article, hybrid PET MR and brain connectivity, where all these concepts are explained. Now I will go to all the approaches in detail. Structural connectivity, as I said before, aims to estimate the axonal pathways. This can be made with the diffusion tensor imaging that is related to diffusion MR imaging with specific MR sequences. We can probe the water diffusion, and as you can see in the bottom figure, water diffusion is high in free water, for instance in the CSF, is isotropic and low in gray matter, and is highly anisotropic with high diffusivity in axonal pathways in white matter. This means that the water is follow some directions, and by studying such direction we can infer axonal pathways. As you can see, this is a schema on an axon. We have a free diffusion along the axonal pathways and restricted diffusion transversally. Therefore, by acquiring at least 6 different directions of diffusion-weighted images, here you can see some selected slices at 6 different directions, with computer-based pipelines, processing pipelines, we can infer a whole brain streamline tractography. Here you can see the axonal pathways. Colors are related to the main direction of each pathway. And with this description of axonal pathways of the brain that is estimated with just at least 5 minutes of magnetic resonance without contrast medium injection. This is very important for clinical translation. And by studying axonal pathways, we can estimate the so-called connector. On the right, you can see a brain parcellation. You can see different colors assigned at different cortical regions, the so-called brain parcels. And for each pair of parcels, we can count the fibers going from connecting two parcels. With this estimation, we can get a matrix that is a symmetric matrix, since we are not looking at direction. And such matrix is a matrix that representing the brain structural organization, and such matrix can be studied with some typical graph theoretic approaches. This is a graph matrix. In fact, there is a strong development of the application of network analysis to brain connectome as estimated with structural connectivity, but also functional connectivity, as I will follow in the next slides. Here you can see that you can study network topological properties, such as the degree of the graph, the hub, rich clef motif, or small wordness of the graph. And such topological parameters can give characterization of pathologies. In here on the right, you can see that complex brain networks have been shown to be sensitive to behavioral variability, cognitive ability, genetic factor, experimental task, gender, for instance, and a huge number of neurological and neurodegenerative diseases. And this is very, very important in order and very impactful on the neurosciences. This year we just published the work that is related to structural brain network reproducibility. We published on the Brain Connectivity journal. And in particular, we investigated the influence of different diffusion, acquisition, and stratigraphy reconstruction schemes on graph metrics. We showed that there is, with the proper recipes, there is a good, very good reproducibility of such techniques. But what I want to highlight in this talk with this slide is that the estimation of structural connectivity is a very complex task, since we have very, very variable recipes on the literature on both acquisition and processing tasks. Here you can see that we included different approaches, deterministic, probabilistic, with different parameters and different diffusion models. And this is why there is a huge literature, but we need more reliability in order to do clinical translation. Regarding the functional connectivity is, regarding magnetic resonance, is related to the study of Bald effect. 30 years ago, Bald effect was investigated, was discovered, and it stands for blood oxygenation level dependent effect. And we know that there is a change of the ratio of oxyhemoglobin and deoxyhemoglobin related to the increase of neuronal activity. Therefore, with Bald signal, we can estimate indirectly neuronal activity. And functional MR imaging is based essentially on task-based. Here you can see a typical experiment where people undergoing fMRI is asked to open and close the eyes. And here you can see the activation of the occipital cortex obtained by subtracting open and closed task. With such condition, we can localize the occipital region as a region devoted to the open and closed eyes task. This similar experiment can be made also with EEG and fMRI. So, regarding functional connectivity, the previous slide showed a task-based typical analysis. With the functional connectivity, we perform the board acquisition. Here on the left, you can see the board acquisition. We acquire each brain volume at a TR of 2 seconds in this case, and each 2 seconds you can have a board signal. Therefore, you can estimate for each voxel the so-called time course of board signal, and in resting state conditions, that therefore patient or subject is asked to think nothing, and you can study, as I said before, the statistical dependence between different time courses. In this case, we consider the voxel A, B, C. You can see the time courses, and we can, for instance, as a basic experiment, we can calculate the Pearson correlation coefficient between time course A and B, A and C, and B and C. Here you can see the different values of the Pearson coefficient. In case of A, B region, we have a significant correlation. This means that regions A and B are functionally connected. And for instance, with this procedure, some networks emerges from functional connectivity analysis. In this picture, you can see the so-called default mode network of functionally connected regions. Regarding clinical outcomes, the study of functional connectivity networks has been very, very impactful in literature, and as I showed for structural connectivity, we have a series of publications that are related to neurological, neuropsychological, and neurodegenerative diseases. It is very sensitive, but all the studies are related to group comparison. Regarding the metabolic connectivity, the first example of metabolic connectivity from the 80s is related to the concept of diaschisis, since some neurologists observed that in consequence of a cortical stroke, for instance, the crossed cerebellar regions was hypometabolic, indeed. Therefore, this was related to the anatomical link from both regions. Therefore, anatomical disruptions from anatomical disruption forward hypometabolic pattern. This is very important for neurological studies. For go deeper in this topic, I suggest this review from Carrera and Tonuni, published in Brain. From the concept of diaschisis during the years, from you can see here the work of Horwitz on 84, you can see different approaches for studying metabolic connectivity. All the historical approaches are related to the study of across subject covariance. How it is performed? You have to normalize, especially normalize PET images, and to study the variation of the signal across subjects. For instance, by estimating the Pearson correlation between regions across subjects, similar to functional connectivity, but functional connectivity was across time at the subjective level. In case of metabolic connectivity, we study the covariance across subjects, therefore at group level. And by studying metabolic covariant, so-called metabolic covariant network, some networks emerges that are similar to resting state functional connectivity network, but with some differences. Again, another concept of current interest is temporal metabolic connectivity. Some researchers are interested to study metabolic connectivity at a subjective level. How to perform this? This can be performed by considering dynamic PET signal. Therefore, you have to acquire dynamic PET signal and to study the uptake, in the case of glucose, the uptake of glucose during the time. You will have a time course of this signal, and by correlating the time course across different regions, some network can emerge. This is very different with respect to the typical metabolic connectivity, but should be considered for a comprehensive view of the brain connectivity. Last but not least, we have also effective connectivity. With effective connectivity, as I said before, we want to study the causal influences between neurons or brain regions. And we, in this case, this scheme is kept from a review about EEG connectivity. And the functional connectivity can be non-directed and directed. In case of directed, there are some statistical powerful tools as gradual causality or cross-correlation, but the crucial prerequisite for all these methods is to have a high temporal resolution. Summarizing different concepts of brain connectivity, here you can see on columns related to structural, functional, metabolic, and effective connectivity. I've tried to clarify the concept and the estimation technique. We have that for some approaches, we have a straightforward concept, but a challenging estimation technique. And for other elusive concept, but well-established techniques. This means that, of course, brain connectivity is a powerful tool, but is in a complex framework. There is no direct relationship between different kind of connectivity, and be careful with the conclusions that pitfalls of underlying concept should be always mind. Better mark to understand underlying physiology is crucial in this sense. Just a moment, let me mute to one minute. Sorry. Therefore, you had the opportunity to give a deep look on this slide, and therefore it is we should be careful with the conclusion pitfalls of underlying concept that should be always minded when we compare different approaches of brain connectivity. Here, as a demonstration of the impact of the brain connectivity approaches, in this slide I've collected some recent results from literature that are mainly the most claiming results. Here you can see an article from Nature Neuroscience where journalists said that people are identified from brain activity, or human IQ is linked to variation of morphometric networks, gender differences, and white matter connectomes predicts cognitive abilities at age 2. Or local connectome phenotypes predict social, health, and cognitive factors. Very impactful results, and we should be also careful not only for clinical impact, but also for social impact of these techniques. But how we can exploit multimodality? Therefore, I want to link the first block of slides with the second block of slides in order to understand how we can exploit multimodality. Recalling this slide, we can exploit multimodality by studying different approaches and probing the relationship between different approaches. For instance, our main work on application of multimodal imaging on brain connectivity was related to the study of a relationship, here you can see on the left, between glucose uptake and functional connectivity as revealed from resting state fMRI. In this work, we know that resting state glucose and oxygen metabolism are closely linked, and FDG uptake reflects energy consumption, and that is related for the major part to neuronal signaling. Therefore, we aimed to explore a relationship between metabolic information provided by resting state FDG PET and fMRI in neurological healthy subjects. This work was published 6 years ago on Neuroimage, and during the years it was highly cited from the research community. Therefore, by using PET-MR, this is our research experimental setup. Therefore, subject was in a quiet dark room after the administration of PET radiotracer. Therefore, after about 40 minutes, MR acquisition started simultaneously with the PET acquisition. Therefore, we acquired MR and PET simultaneously. We had to face the different timing of both modalities, since for FDG PET we have an incremental signal during the time. Therefore, after the injection, what I see with the PET scan is what happened during the 30 minutes before, since from the administration to the scan beginning, and is a static scan. Resting state fMRI is a dynamic scan. Therefore, we had first to properly process all the data. Here you can see the processing pipeline. And therefore, we had to calculate some functional connectivity metrics that were comparable with static FDG PET. Therefore, we included 3 different metrics. The first was the regional homogeneity that estimates the degree of synchronization between time series of a given voxel with its nearest neighbors. And in this sense, it could be considered as a local range functional connectivity metric. We have FALF, that is a fractional amplitude of low frequency fluctuations that quantify the amplitude of low frequency oscillations, and is a voxel level functional connectivity metric. And last but not least, a long range functional connectivity metric, that is the degree of centrality, that considers the degree, how many voxels are connected to the considered voxel with the functional connectivity. This is similar to the degree of a node in a graph theoretic approach. As a result, we showed a comparison of the functional connectivity metrics. Here you can see rho, FALF, and degree of connectivity, a comparison with FDG PET patterns. You can see that the patterns are very similar, especially for rho, and this was the first time a simultaneous PET-MR study showed such similarity of patterns. 3 years later, always on Neuroimage journal, we showed similar patterns in a similar experimental setup on Alzheimer's disease patients. Our results was that the relationship between FDG PET and fMRI metrics is reduced in advanced MCI and Alzheimer's disease patients. Here you can see on the top left FDG, typical FDG apometabolic pattern, on top right FALF, rho, and results from independent component analysis. And this was very important, but all these approaches was very important for literature. They were the first results released in literature, but are still data-driven approaches. And we are currently working on hypothesis-driven approaches of energetic cost of functional connectivity. And this is my main research interest for the current and for the following years. Another clinical application is in DOC patients. DOC stands for disorders of consciousness. With my colleague Carlo Cavaliere, a neuroradiologist that is responsible for the unit of Naples, we acquired PET-MR on disorders of consciousness patients. And here you can see 2 works seated on this data. The first, on the left, was a study on the variation of brain connectivity among different time points. Here you can see on the columns we have MCS and awake patients with different condition of disorders of consciousness, and we demonstrate the variability of brain connectivity during the time in these patients across time and within acquisition session. On the right, you can see another work where we propose also a nice picture representing the connectivity of the brain, and as well as also the PET uptake. This kind of graphs are used to have a comprehensive view about multimodal characterization of brain connectivity. Each region is a brain parcel, as I shown before in the case of connectome estimation. Another hot topic that is related to the relationship between different connectivity approaches is the structure-function relationship. We have a topological basis that is indeed the axonal pathways, but on this topological view that is indeed the roots of the connectivity. On these routes, some patterns of functional connectivity emerges. But such patterns are not congruent with anatomical and functional connectivity. The link is not perfect, and there is a great interest in unveiling such what is the link between structure and function in order to understand the brain organization. And we are interested in studying the brain as a dynamic system and studying the emerging properties of this system in order to explain the structure-function relationship. Let me also share with you this opportunity. Since 10 years ago, Human Connectome project from United States started, and I think that 2 years ago, it released very, very useful data for playing with the brain connectivity. There are indeed 1,200 subjects acquired. You can get all the data by registering on the Human Connectome project website, and 1,200 subjects that were acquired with magnetic resonance, task fMRI, resting state fMRI, structural connectivity, task fMRI in different domains, also behavioral characterization for all the subjects. Some subjects were acquired also on 7 Tesla with a high spatial resolution, and 100 subjects also with MEG and EEG, also resting state. Therefore, these data are very, very, very important in order to study brain connectivity, also where you are not linked to a clinical site. Therefore, also engineers, physicists can play with brain connectivity by downloading such data released by the Human Connectome project. And here you can see that the overreaching purpose of the project was to acquire and share structural functional connectivity of the human brain data, accelerating progress in the emerging field of human connectomics. And this is what I'm talking about today. So this is I think the last slide, yes, with the final remarks. I showed that multimodal imaging provides a comprehensive view on brain connectivity, and brain connectivity has enormous potential for clinical applications and beyond, as I shown also with societal implications. But accuracy, reproducibility, and reliability should be demonstrated at subject level for an effective clinical translation. But be careful, since brain connectivity approaches are very different and probably they are completely unrelated with each other. And in order to fix this point, in order to highlight this point, here this is a paragraph from the abstract of the article of Horwitz, that is one of the pioneers of brain connectivity. The title is The Elusive Concept of Brain Connectivity. I suggest you to give a look on this paper, that is one of the pioneering papers on brain connectivity. He said that the terms of functional and effective connectivity are applied to quantity computed on types of functional imaging data that vary in spatial, temporal, and other features. and employing different computational algorithms. There are in literature very different recipes in order to process such kind of data. And we need to target our analysis on good choices. Therefore, until it is understood what each definition means in terms of underlying neuronal substrate, we have to consider the underlying neuronal substrate, comparisons of functional and or effective connectivity across studies may appear inconsistent and should be performed with great caution. And this is the last slide, just to promote the PSMR conference that will be held in Elba Island in Tuscany next spring. And we are arranging a session about the topic of multimodal imaging and brain connectivity, and you are welcome to attend. Thank you very much for your interest. Thank you.